So, I am not a mathematician, I am an experimental biologist, and I have, as Sitabra mentioned, I have very different background. So, I got trained and as undergrad as a chemist, then I moved to East and working on translation initiation, and but somehow this development, the question of how systems develop, that's intrigued me. So, I moved to flies, and now I am working with worms for some time. So, what you see is a very simple clock, but the most interesting thing is how do you get there from this pile of tools? That is the question that we are all interested in. So, it is a reversible process. You can assemble, disassemble, you can assemble it in a different way to get to this thing. So, but when we talk about brain, so this is a similar system. So, we want to understand how exactly these precise connections form, and how, and also another interesting part is how these connections do not form, and how we go from here to there, where we can change precisely how to alter some of these connections, but not all the other ones. And if you think about, like in many disease conditions, like specifically in schizophrenia, where you have like hallucinations, and many of the causes, underlying causes of those things are these miswiring. So, when we talk about brains, there are three different types of connections that we are interested in. So, we talk about chemical synapses. So, mostly, like we study, when we look at the neuroscience textbook, we study the chemical synapses. These are the by far the most important components of the neural connection. So, where the signal is received by this cell, and they will send these packets full of neurotransmitters that will be sensed on the postsynaptic side, and depending on what are the receptors, they will give different sort of signal. So, it can be inhibitory, it can be excitatory, it can be like different time gaps can be there. Also, there is another type of synapses called electrical synapses that does not need these neurotransmitters or like receptors. They are direct coupling between two cells through these channels called gap junctions. So, each of these synapses consists of thousands of these channels, and they are allow direct transfer of ions and molecules. So, I will cover this thing in the last lecture tomorrow. We are mostly interested in this, but let us look at how these but one thing I want to mention, these two are hardwired connections, means they are direct coupling between two neurons. You also have non-hardwired connections that are equally important. They are you cannot see them by looking at the EM section, because they can also act over a distance, which are like monoaminergic, like and neuropeptidergic synapses. They also can act as a neuromodulatory. So, it can modulate the activity of these synapses. So, why do we need connectome? So, we from last talk from Professor Bapi, we also understand that there is we need to understand the connectome and all that, but how it started. So, a connectome as its base, it will give us a map, so that we can study how the genes specify these connectomes and how the nervous system can generate behavior as its entirety. Okay, and how does it change? So, meaning that if we know the map, we can understand how it precisely getting altered. So, at this time, when we look at this problem, what we have in our hand, if we deal with human brains as Professor Bapi was doing, and so it is a very complex system. So, here you have 86 billion parts or 86 billion neurons, and equal amount of glial cells, and they are connected by trillion connections or synapses. So, how do you precisely understand how these neurons are connected to each other? Okay, so you need a simpler system, and this is, we cannot start a neuroscience talk without mentioning Ramon y Cajal. So, he is the father of modern neuroscience. So, this is a picture of Ramon y Cajal, a hand drawn picture from Ramon y Cajal. So, this is a Purkinje neuron in human brain, in the human cerebellum, and you can see this very complicated structure. And so, each of these neurons are just like a tree, and one single neuron here in our brain can connect to 1,75,000 connections. So it's incredibly diverse. So you need a simpler system. And so since it's the first lecture, I'll take a 1 minute to give you a historical background. So before Ramani Kahal, so there was a, so all of you probably heard about Golgi, like Camillo Golgi. So there was there were two groups, Golgi's group and Kahal's group. People before thought that in our brain it's a neural net, means that there are no cells, there are just wires that are connected to each other. Kahal actually proposed that it's actually cells that makes this connection. So before that, the neurons theory were not present there. So it kind of sounds bizarre at this point, but it was that time. So Kahal, nobody believed Kahal. So he had a tiny microscope. He developed a staining protocol that was previously identified by Golgi, and he did the stains and sectioned them, took these pictures, hand drawn them, like thousands of this, and you can actually still see in some of the science galleries these pictures, and it's incredible. And he took those pictures and traveled all around Europe to convince people that this is actually true, that in our brain there are neurons that are making this connection. So it sounds bizarre, but it started from there. Okay, so let's go back to our topic. So how do you understand the connectome? We need a simpler, so that time, like people started to understand that we need a simpler organism. And before that, to give you context, like around that time, like around 1960s, there was another group of people, two scientists who studied kittens. And so what they did, they took kittens right after their birth, and they covered like one eye of those kittens. So it's called monoocular deprivation. And then they looked at what happens to those kittens when they develop. And so those kittens who had developed with one eye covered, they had like completely different neural wiring in their optic lobe versus the ones that develop properly. So we knew that there is a connection at that time, even in higher vertebrates, in kittens, but it's too complicated to understand what actually the ground state and how it changes. So it again points out to the same fact that we needed a simpler organism. So at that time, this guy came, Sidney Brenner. He's a pioneering guy. So he's one of the pioneering person who in 1960s decoded the how the codons are organized in the human in the genome, and the triplets and all these kind of things. He's one of the founding fathers of the modern molecular biology. Okay, so he's thought that we need a simpler organism, that the contexts were the same, that has to have a genetics, a behavior, and is malleable. So he chose this simple organism, C. elegans. So some other people were doing C. elegans work at that time, but he looked for many years, he looked for small invertebrates that has good nervous system and can be fixed and stained to see the nervous system, just like Kahal, and it can also have a simpler behavior, simple reproducible behavior. So these animals are 1 millimeter in length, like they're adults ones, and you can see this is a zoomed in picture. There are two sexes. One is hermaphrodite that produces their own sperm, and also it has males. And naturally occurring population, there are 0.05% is the male, and it grows on petri dishes, and you can put agar and bacteria lawn, so they feed on bacteria. So you can find them, these nematodes, in rotten vegetables or rotten fruits normally, and they will feed on the bacteria that are growing there. And they also have like fixed number of neurons, only 302 neurons, and I'll try to convince you, even we don't understand how these 302 neurons make circuits and organize. And they make very precise behavior. So here you can see the worm is moving, it has a very gentle, like sinusoidal coordinated movement, and if you take an eyelash and touch them on their head, what this is video shows, it goes back. So it has an incredibly sensitive nervous system that can sense like this low threshold touch with an eyelash versus if you poke them with a metal rod. They can actually distinguish, there are two different circuits. So it's kind of like for us in human context, if you tap somebody, you need to sense that there is a touch, and also is a different threshold touch, versus like that when you when a piece of cloth like on your arm. So you have to have like two different types of sensory thing, and that is what worms also have. They also have memory. They uh, change like behaviors according to their context, internal and external context. So it's a very good system. So how to make a connectome with this? So this is a pipeline that came from a modern protocol that is in 2018, and this pipeline was exactly the same in Sidney Brenner's time as well. So you have to take the worm, you fix it in a mold within a resin, and you have to cut it with a like a salami slicing it, and each section can be 90 to 70 to 90 nanometer range, and you can take pictures of these sections in piece by piece, and you have to like stitch these images at high resolution images to get the whole thing. So here you can see that this is one section of this nematode, and here inside this dark part is the pharynx, and around it there are neurons, and at the edges there are muscles and hypodermis or the skin. And you can do the cell ID by tracing in different ways. You can do machine learning and all sorts of things, and then you can also have like now we also have machine learning to annotate like synapses, and then you can get a topology reconstitution from these things. But they didn't have that in 1970s. Yeah.
Since there are only 300, we do it by like manually.
Yeah, that's what exactly they did it. But I'll tell you like what is the problem. So at that time they didn't have any of these things. So what they did like they initially thought that okay, let's build a operating system, let's build a computer, let's generate these algorithms, and we will do it this way. So they were way ahead of their time. So they built this computer. It's called, they named it modular 1 computer, and you can see this whole wardrobe thing is the CPU, and it had a memory of 64 kilobyte, and this entire box, this giant coffin type of thing, was a hard drive that had 22 MB storage space. So they tried to solve this problem using this computer. So that was the best they could get. Obviously it wasn't enough, so it could not solve this problem. So but this guy is standing here is called as John White. He is the pioneer of this whole connectome business with Sidney Brenner. He was a graduate student in Brenner's lab. So what they did, they printed all of these things, all these images on 22 by 16 inch glossy prints, and they hired a technician, Donna Albertson. So she was very good at puzzle solving. So that was the criteria. So at that time they also had like a very good invention. So that was the first time when these colored marker pens were invented. So now they could get like different color pens, and they painstakingly with a microscope they labeled all these neurons one by one in different colors. And they can also see here, you can see this dark patch here, that is the presynaptic density where all the neurotransmitter receptor field packets are sitting to be released on the other side. And here also you can see the neurotransmitter presynaptic density, and these tiny vesicles here, if you can see them, they are the synaptic vesicles. Okay, so now, so what they did, they took this, printed these things, and they got this other sections on these films, these negatives, and so they took the picture and they projected on the another one, next one on top of it, and they layer by layer they built the whole connectome. Okay. Yeah. So they stained with the EM, like with osmium tetroxide and like regular way how you do it there. But there is a sad story, but that time the regulation was not that great. So all these osmium tetroxide they used, they had uranium contamination. So many of the people who worked in this project later on had cancer. So that's a sad story. But now to replicate that kind of staining is very difficult, because now we have better osmium tetroxide which doesn't stain this way. But people have devised like different way of staining it. So they are like osmium tetroxide stained. No, they naturally occur as dense vesicles because they have more proteins, they're more electron rich, so they will stain more, and when you take a picture, they will appear dense.
How would that be different from cell junction?
That's a good question. So cell junction won't have this presynaptic density, but the problem arises when you're talking about cell junction, trying to distinguish between cell junction versus gap junctions. That's a big problem. So their cell junctions will have like a, if you look at the measurements, how far two cells would be, that is different than gap junctions. And also that how the membranes are juxtaposed to each other, that will also be different. But it's very tricky. So if you don't have, so oftentimes what happens, here you are slicing the animal. If you have minimum distortion, you will lose that clarity to call that whether it's a cell junction versus a gap junction. But most of the time what happens, like it can be a problem for one section, but generally these features run for several sections. So you can check whether there is any similar features are appearing on the next sections or not. And luckily for most of the nervous system in our case, or also in Synerhabditis case, they are left right symmetric. So you can also go to the right side and check whether you get a similar feature or not. So you have to do a lot of cross checking, but it's still very difficult. It's a very good point. So they published this paper. It took them 15 years to do the project. So what Akshay was saying, that can we do it manually? Yeah, but it will take another 15 years. Okay. So and it had 450 pages. The entire volume of the Royal Society journal was dedicated to that. And at that time, in 1986, it took 17,000 pounds to publish this whole thing. So, but it gave us the first ever connectome of an entire nervous system. And you can see now that all these neurons, they're named arbitrarily, like how they were naming it. So, so this R stands for something, sometimes right, left. So that's they to distinguish between this one neuron versus other, they named it and the name stayed. So here you can see how they are connected and which way the direction is going. At least in case of chemical synapses, if you see a presynaptic density and the you can call it like a presynaptic site versus postsynaptic site, you can put the direction of the arrow. But in case of gap junctions, you cannot say that. And so it gives us lot of this information. So these are different classes of neurons. So the triangles are generally mean the sensory neurons. The hexagons are that's how they differentiate it. So motor neurons are one type, the interneurons are another shape, and the sensory neurons are another shape. But sometimes it's also confusing because some of the neurons, like sensory neurons, generally we call a sensory neuron that only receives information and just transmits. But some of the neurons are present at the sensory interneuron junction because they can be both sensory and interneuron. Also the interneuron motor neuron. So that's how some of them also has different shapes. Yeah.
With just the EM sectioning, how did they identify the function of the neuron?
That's a good question. We don't know that. So at that time it was not known. So that is the next question. So now we have the connectome. Now how to put function into there? Okay. So then this guy Marty Chalfie, he was a postdoc in Sidney Brenner's lab. He came and he tried to understand how these worms are sensing this gentle touch. So later on he figured out, because he was at that time he had the tools to look at what are the neurons, what could be the possible connections. So he took the worms, put it randomly, mutagenized it, and painstakingly take those worms and touch them one by one, and found a lot of mutants that are called MEC mutants, means they're mechanosensory defective mutants. And so and many of the channels and how the mechanoreceptors that he discovered in worms in 1986, we still work on them, and it's the same type of receptors, mechanosensory receptors, that we also have in our cells. So it's highly conserved. and the basic principle also stayed the same. And he later on, since the connectome was published, he could also look at how these neurons, there are 6 neurons in that circuit, 2 in the head and 2, like 1 here, 2 there and 2 in the posterior part, that dictates how the worm is going to sense. And that is the sensory neuron, mechanosensory neuron, how they will channel to the command interneurons or the premotor interneurons, and that will generate the bimodal behavior. And we can also solve these problems, but we still do not understand the whole circuit, the functional circuit. I will talk about it in the last part. So, okay, so this guy is also important because by doing these things, he at that time, like in around 90s, early 90s, he discovered that there are some people who cloned a gene that gives the fluorescence protein. So, he is the first one to start using GFP to mark the neurons, and for that he got the Nobel Prize for using GFP. But apart from that GFP contribution, he is the first one to give us an entire circuit to say how animals interact with the environment. Okay, so this is the circuit that we know coming from Marty Chalfie's work and other people's work over the years, that there are these sensory neurons at the top, you have these directional arrows that are chemical synapses, and the bidirectional these lines that are electrical synapses that are heavily coupled to premotor interneurons or the command interneurons. And these command interneurons, so these are sensory layer, this is the command interneuron layer, then you also have some computing interneuron layer, and these are motor neurons, and that talks to the end organ, the muscles. So, here you have you can think about a logical way of interpreting the circuit. So, if you touch a worm in the head, it needs to go back, so it will talk to the backward command interneuron. And so this is the forward anterior touch sensing sensory neuron, and this is the backward command interneuron circuit. So, it is going, this sensory input is going to the backward command interneuron and then backward muscles. But when you touch in the forward backward, it goes to the forward command interneurons and it goes back forward. Yeah. Okay. So, also you can also think that while the worm is moving backward, it not only triggers the backward motion, but it also has to stop the forward movement. So, there are also inputs, negative inputs to the forward circuit that are coming. So, this is a basic principle. And so, but how to improve on this, because that time they did the connectome manually, there were a lot of the microscope were not there, so there were a lot of misses. People realized that there could be some more and more misses that we need to identify, because later on functional studies said that there should be a synapse, but the connectome does not say that. So, these guys took the same sections, same pictures that John White and Sidney Brenner made, digitized them. Now they have in like 2000s they have the computation power. So, they did it and they digitized them, they devised some algorithms to orient them in a proper way. Now they have those tools, so they could build it. But still it took them very long time, because I know this project very well, because this lab was right underneath where I did my PhD, and this guy is my PhD postdoc advisor. So, I know that this worked very well. It took them incredible amount of time to devise this. So, now what they did redid the entire hermaphrodite connectome, and they also did the male connectome. Now, what you can do, you can actually compare them, how these sex shared neurons, meaning that which are common between hermaphrodite and males, how they are connected, whether is there. So, in let me rephrase my wording. So, in C. elegans you have 302 neurons in hermaphrodite, and in male there are 384 neurons. So, around 9 or 10 neurons in hermaphrodite that are specific for hermaphrodite, that are required for egg laying. and the male, the extra neurons that are not there in the earlier development, but they born later on before the sexual maturation, they are required for male mating behavior. So, the majority of the nervous system is shared between male and the hermaphrodite. So, the question is, the sex shared neurons, are they exactly forming the similar things, or there are inputs from the sexually the reproductive circuit on top of the shared nervous system, so that that can also be changed. Okay. So, they found that the basic principle of the connectome remained the same, but lot of inputs came from the sexual circuit on the interneuron or modulatory neuron circuit, so that it can also generate different behaviors. Okay. So, I will just give you like some very brief part of it. So, if you took the fraction of chemical synaptic ages, means the synapses, and the nodes are the neurons and the edges are the synapses, and the age weight, if you see that hermaphrodite versus males, so some of the male connectomes, specifically this connection is a specifically required for male mating behavior, is incredibly has like lot of age weight. Means how do you calculate the age weight? Because you take the sections, and if the age weight is these numbers digital, these numbers are meaning that how many sections, consecutive sections, this synapses span. So, if you take a section like from 1 to 2 to 3, so this is a, and if you see that this the synapse is still spanning like these 3 sections, it means this has a age weight of 3, meaning that here this synapse spanned like 200 sections. So, that is a huge synapse, and you can say that okay, this huge synapse means this is like functionally also strong, but it does not. So, it is a correlation. Again I am saying that there is no functional test you can do from the connectome. So, so you can see the majority like this chemical synapses has overall has a tendency to be stronger in males compared to hermaphrodites, and the gap junctions, they are pretty similar. But if you look at the weight, yeah.
You said the longer the synapse, stronger the connection.
That's a correlation. We don't know that. There is no functional data to it.
But wouldn't you expect it the other way? Because smaller the gap, more efficient the communication.
No. It's not the gap. So if you take the worm, if you are cutting it, right? Like 1 section, 2 sections, 3 sections. The longer the more number of sections means the synapse is present like wider. So it is like a bigger structure. Are you clear or? Okay.
Will it depend on how you cut the?
Yeah. So if you imagine this is a worm, okay, so you are cutting it like this, and most of the neurons are spanning like this. So if a synapse is only this big, you will see it only in 2 sections, but if the synapse is very big, you will see it in more sections. So that is a proxy. You can say that this is a huge synapse means is functionally important, but it does not tell you whether it is actually functionally important or not. That you have to do some either electrophysiology or some activity assays.
Okay, but the communication would be more like less efficient between a longer synapse.
No,
This is really diffusion to fill the bridge, fill the gap.
No, it's not the gap, meaning that you have neuron A and neuron B that are running next to each other, okay, and they have like physical proximity all along the body. Now you can make a connection only this much, or you can make a connection much bigger.
Oh, okay.
So the distance is not different. So they have physical proximity, but how long you can make the synapse. So the bigger synapse means that you have more number of like neurotransmitter field vesicles could be present there. So when you stimulate it, the stronger signal can pass. So that's a proxy.
You talk about the Like how wide?
How big the physical structure is.
These are the two neurons.
Yeah.
How wide is this
So,
direction?
so one thing I think is confusing, because when we talk in the normal sense, we talk about this is the synapse.
You're talking about the height, not the width.
No, so this is the synapse that we generally study in the textbook. But there are many cases in invertebrates, vertebrates, in our brain also, there are synapses called opposite synapses. Means the two neurons form synapses while they are passing each other. Means that it's not at the end of the neurite. So here there is a neurite, here there is a neurite, there, but while the neurite is passing, they are forming synapses all along the length. Okay, so I think you are confusing between this to this. Yeah, so this is very important distinction, but it's not, it doesn't mean that the C. elegans doesn't form the end point synapses, but they do form this one. In our brain also there are many opposite synapses, especially in the interneuron level. It's clear now?
Yeah.
Okay, but it still doesn't guarantee you that it will transmit faster or there will be more volumetric transmission. Like, so that we have to test, but it has a capacity to do that. Yeah.
So these opposite synapses don't have myelin sheaths?
So no, so myelin sheath you need because you need to
Transmit it over longer.
transmit it for a longer distance. So these are, they're not too long, so that's why you don't need myelin sheath. So for a short distance you don't need myelin sheath because you don't need to insulate. The other distinction that you have is, I didn't want to get into that, but okay. So now in our brain we have, like, millions of neurons, right? So you can dedicate some neurons for this task, some neuron for that task. You don't need to, like, there is no overlap, and that's good because there is less chance of having error between, like, confusion between two different stimulus. But in C. elegans or in lower or invertebrates, they do not have that many options. So they have to do all of it with 302 neuron. How do you do it? Because so they don't have this digital type of transmission. So in our case either is 0 or 1, but in C. elegans is more like an analog transmission. So means that if you have a cell body, when the signal comes, in our case it travels all along the axon. So you have the axon potential, but in C. elegans you don't have the axon potential most of the cases. So what advantage it gives, it restricts the activity to a certain zone, so it doesn't travel. So what you can do, if you, and that is also true in some cases in vertebrate brains, but it's more risky to have it in vertebrate brains because it's more, you can have more overlap. So you have more neurons, so you can afford to dedicate certain ones to certain places. So I think that's why we have it separate. So what you can do is, like, suppose, like, there is a stimulus A that is coming, only this region is functioning. At the same time, another stimulus B can activate this part of the neurite, and two computations can happen at the same time using a single neuron, because there is no transmission passing from this place to this place. So that's why it's also, like, so it's kind of misnomer if when we talk about the invertebrate, the nervous system is simple, because the computation may not be simpler, it's just smaller. Okay, let's go back to the original thing. So if you look at that in the connectome, if you just look at the hermaphrodite, yeah.
Is it something similar to dendritic computations that happen in the
Somewhat similar, because now we know that in dendrites different branches can have different computation. So I think it's very similar to that. Yes, and you can also have, like, you can also have cell body doing something, an early part of the axon doing something and later part of the axon doing something else. So yeah, so that is precisely true. Like, dendrites, you can have different branches doing different things. Yes. So if you look at the gap junctions, just that there is an incredible amount of diversity in terms of how much how big they can be or how small they can be. But if you look at that here in this graph, the majority of the weight of the connection is being done by synapses that are smaller. Okay. So, there is no precise correlation about the strength of the synapse, the functional importance of the synapse versus the weight. So, it is, if you think that C. elegans, it has a I will show you the picture here. So, this is the majority of the connectome, that is connecting all the sensory neurons, the muscles and all that, and this tiny bit of connectome here, separate connectome, is coming from the pharynx. So, there are pharyngeal nervous system that only talking to each other mostly. So, they are required for precise control of the feeding behavior, but they also need to be coordinated with the somatic nervous system. So, there is a, I think you can't see the line here, there is a single line that connects between the somatic nervous system to pharyngeal nervous system, and it is a gap junction. It is a very tiny synapse, but if you take it out, the connection between the whole somatic nervous system to the other part of the nervous system is completely gone. So, the weight of the synapse and the functional importance, it does not go hand in hand. Okay. So, there are just some list of text. So, what is the importance, what we gain from the synapse? So, after we got the connectome, nearly all the neuroscience studies benefited. So, you start with the connectome, you build your hypothesis based on that hardwired connectome, and then you study it, and it provided a map to tell us how information can flow to generate behavior, but it does not guarantee that the information will flow through that route. So, it is kind of building a road network. So, that tells you that a car can go from one place to another through these roads, but it does not guarantee the car will always go from that road. So, this is the difference between functional network and the physical network. Okay. And but this is just not the worm. So, when we learn about the C. elegans connectome and the network, we also learned a general principles how nervous systems can be arranged in a functional circuit. Okay. So, after C. elegans, now we have connectome of some part of the Drosophila brain, some part of the mouse, and also we have different nematodes, that is Pristionchus pacificus. So, this is a predatory nematode. So, they can feed on C. elegans also. We have a full connectome of that, and now you can compare that how two nematodes that are evolutionarily separated by millions of years, how their blueprint is stayed conserved or diversified. And also you have other some other invertebrates, as well as a small chordate, the Siona intestinalis, the whole nervous system of that. Okay. So, but unfortunately, except Siona, none of the other nervous system has information about gap junctions or electrical synapses, because of the problem that you mentioned, that is very difficult to determine which one is just membrane proximity or an adherence junction or a gap junction. It is very difficult unless you have a very precise sectioning, and that is very difficult to do in higher order brains. Okay. So, now we have much better understanding how these ones work. So, you just just look at this movie. This is the nose of the C. elegans, and here you all have the sensory cilia, and this is the reconstruction based on this EM connectome, and this is the structure of the sensory cilia that are present in the C. elegans nose. So, this is the outer silhouette is the body, and this is what you can see, that all these sensory cilia are intertwined with each other, and they are kind of required, these precise structures are required for sensing the external environment. And if I can replay this movie, you can I'll point out like in one cilia here. So look at these cilia that are like precisely like this lot of lines. This is actually thermosensory cilia, and when we look at the physics like school physics textbooks, like how do you have like a greater exchange of heat when you if you have like lot of surface. So this is a thermosensory cilia, and it has exactly the same principles for heat exchange, and this was possible only because we have the connectome. Yeah.
When you say cilia, you mean neurons?
So this is the end of the neuron. So these are the cilia. So where you have the dendrite, then these cilia are exposed to the environment where they're sensing the receptors. They have like all the sensory receptors where you have like heat, cold, food odor, sodium chloride, or like whatever we have. So that's also extensive sensory neurons. So, and this is like when we have in our nose the olfactory neurons, they all have very similar type of receptors. They also have olfactory receptors. Some of them are also stimulated by light. They have like primitive opsins. So these are like all very similar stuff, and these structures we know exactly how precisely they are maintained. They're maintained by glia, like in all these precise like in intricate details we can only come up because we had this connectome. So that's the importance of the connectome. But not only that, when we knew about the structure and also like functional connectome, some of the people actually devised robots based on the C. elegans nervous system. So these are not programmed. So you can see like you can build robots with Legos, and they're not programmed. They have like their own mind, and you can just people try to do that based on C. elegans nervous system, and it works. Okay, so but the question is, how do you change the nervous system with development? Because you have to think about like so that happens in our body as well. So C. elegans that goes through 4 defined larval stages from L1, L2, L3, L4 to reproductive adult, and by the course of this development the animal grows 5-fold. So this is around like 1 millimeter, and so it goes from like 0.25 millimeter to 1 millimeter. So there is an enormous amount of stretching going on. They also have some new neurons coming in. They also have environmental fluctuations, so they need to learn how to adapt to the environment. Then also you have like some adult specific behaviors by reproductive behavior. You also have to like memorize some internal and external cues. So like you can, I'll give you a very quick example. So when you are hungry, you are walking in a village or in a street, and you can see like there are some street vendors that are selling some spicy things. It would be very attractive to go and eat those things, but after a heavy meal those same odors can be very repulsive to you, so you don't like to go there, okay, because your internal state has changed. So C. elegans also learn to do those things, because if you feed them they will be repelled by this certain type of odor. If you starve them for very longer, the same odors they can come back and they get attractive. So these are the things that evolve over development. So how does the nervous system mature and evolve? So that's another thing that we can learn from C. elegans. So what people did, like there's a group in Canada, they did exactly the same thing. They took worms and they precisely did the connectome at every different stages. So they have only map until the head of the worm, so we don't have the entire body, but the head is complete. And here you can see that this is the ganglia. They have like a volumetric projection of this ganglia, and you can see the different types of neurons are like, or the end organs are projected here. So the green is muscle, so the muscle is here. You have the sensory layer that are projected in the neuropil, which is the brain, primitive brain of the worm, and here are the connectome. So, you can see that there are different animals. So, there are 4 animals that are at the same stage of the development, and you can see that the overall architecture of the network stays the same between different individuals. So, there is it's very precise. So, it does not change from individual to individual, okay, the basic framework. And if when you go from L1 to L2, you start seeing these colors are getting darker, means these new connections are forming and the older connections are getting thicker. And when you reach from L1 to adult, you can see that so many different things have happened. So, it gave us a blueprint to study how nervous systems evolve over development, and what is required to preserve and what is not required to preserve. Okay. So, if you look at, like, these are some statistics. If you look at the neurite length is growing 5-fold, but if you look at the contacts, although the neurites are growing, if you see that this neuropil is very tightly packed, there is no place to jiggle around here. So, the precise contacts remains the same, it does not change. So, the neurons are not changing their neighborhoods, the neighborhoods remains the same, but the neurites are growing, and as the number of synapses are also growing, kind of linear relationship with the neurite length increase. Okay. So, but the synapse densities are not changing, meaning that the new neurons are coming, but they are just new neurons are generated over the whole length of the neurite. It is not like one region is getting properly extensively dense. So, you can also, like, there are many cases, like, one connection, many synapses are stronger, there are weaker synapses, and you can also see that synapses per connections are increasing. Okay. So, this is, if you look at this thing, this last part, the probability of new connections is increasing over that, if you have more contacts. So, the more contact the neurons have, the more probability, the higher probability is there to form connections. So, which is, like, not a huge surprise. But what is the theme that came out is that at birth you can make, like, the separation between sensory layer, interneuron layer, motor neuron layers were not that great. So, here you can see roughly you can say that this is sensory neuron, this is the modulatory neurons, means, like, it controls like how sensory neuron will talk to interneurons. These are the interneurons, motor neurons and muscles. So, what happens over development, there are certain neurons, especially the interneurons, that takes up the most of the computation. We call them hub neuron, that is present in all sorts of networks. So, these hub neurons get stronger, precisely that the input to the hub neuron gets stronger, but not the output. So, now you can see from earlier development to the adult development, the input to the hub neuron getting stronger, but not the output that much. Okay. So, yeah. So, that hub neuron is getting stronger, and the so that's, as I mentioned, that selective input gets changed, but not the output. But also you have, like, the similar sort of prevalent uniform variability. Some of these modulatory neurons are connected to similar sort of connections, but also you can see that there is a higher, like a more increase in the feed forward bias, meaning that what we call feed forward bias is when signal is flowing from sensory side to the downward side, meaning it's the interneuron side or a motor neuron side. If the feedback loop is when interneuron gives back signal to the sensory side to modulate their behavior. So, most of the time this sensory the feed forward loop is getting increased, but not the feedback type. And now you can start seeing some sort of modularity. The colors are not visible, but you can start seeing that this is becoming a new module, this is becoming a new module, and so on and so forth. Okay. And some of the blue arrows, that are new connections that also arise here. if you look at like one single circuit, you can see that this is what happens. This is the sensory side and this is in the adult. So now you can see like a lot of new inputs that are getting stronger from sensory side to the interneuron side, but not the reverse inputs. And if you look at this neuron AIA, the names are not important. This is a hub neuron because it talks to many of the connected places, and this is an interneuron, and you can see that precisely this neuron input to this hub neuron is getting very stronger. So these neurons becoming much more central piece of the connectome to compute the behavior. Okay. So, and if you look at the statistics, this is the amount of stable connection, and this is, we call it variable connections. Okay. So, variable connections means that are variable between individuals. These are not like variable between different developmental stages, and these are developmentally new connections. So, although the animal is maturing, the majority of the connections that are still retained are the stable connections, but there are some developmentally dynamic connections. These connections precisely occur mostly in the modulatory circuit. So, that modulates this the basic framework. Okay. So, you can still see the number of synapses. So, these are the connections, means the contacts, but if you look at the number of synapses, it is still predominated by this, dominated by these stable connections. The variable connections are there that are will give you like individuality, but also like this new developmental connections are there, but the majority of the connections remains the same. And this is a similar thing that is presented in a different way. So, you can take that sensory neurons that are over. So, this is the proportion of developmentally dynamic connections. The higher it is, the more dynamic they are. So, you can see that their feedback to the modulatory neuron is higher, and also the forward bias is there. And you can see that number of variable connections, these variable connections means they are different between different individuals. They are mostly predominated in the interneuron side, means like where, so the sensory apparatus and the end organs like the muscles and the motor neurons, that are basic principles of the basic framework of the connectome that remains the same, but the interneurons, how they are connected to each other, that will generate like diversity in the output. So, that is different between different individuals. So, that gives the individuality. So, there is not much surprise is there. So, here is very similar. I will quickly go over it. So, this is the feed forward loop connections that are from the sensory neuron to interneurons or the interneuron to motor neuron side are increasing, while the feedback loops are getting, feedback connections are getting decreased. So, overall like you get stronger sensory input. People have also done functional assays where you can put like a different concentration of attractant to worms, and if you see that, suppose like C. elegans can be attracted to one particular type of concentration of odors in L1 stage, they will be able to sense a much lower concentration of odors in the adult stage, because now they have a much stronger sensory circuit. So, they will sense better. The other important thing that arises from here is this modularity or the circuits. So, in the earlier stages it was not very clear. So, if you look at the L1 or right after birth, there were basically two different circuits, like this is, you can call it like a sensory circuit, and this is like a motor muscle circuit here. But then L1 you have like sort of like a sensory circuit, like a head movement circuit, and the motor output circuit for the body bends. But when in the adult you started seeing this different fragmentation of this more higher order modules or more higher order circuits are getting evolved. Okay. So, I will take like 2 more minutes here. So one thing I talked to you about, like how it this circuit like evolves from different developmental stages. But what other interesting thing is, like when you starve a worm population, it goes into a hibernation type of stage, is diapause stage. What it allows, like this life cycle in a normal temperature can be completed in 3 days. But when they sense there is starvation, there is like the environment is not conducive enough to go through the life cycle, they go into this hibernation stage, we call it dower stage. In that stage, they can actually wait for 3 to 4 months. So they are so tough that you cannot like disrupt their cuticle. They are thin. They are like this is a dower larvae, and this is a happy C. elegans that is feeding on this cloudy E. coli culture. And these ones, they stay longer, quiescent. They are preserving their energy, and they also do some very precise specific behavior. So here you can see that these worms are actually literally standing on their tail and waving on the in the air. So what it does, they are trying to make them bigger. So imagine like this is a place where there is no food, they are just waiting to be transferred to a different place. So they are trying to make them bigger, and what happens is like when a insect is like crawling over them, they have a better chance of like getting attached to their leg. So they will take a hitchhike to a different place where might be there is a better chance of survival. And also there are a lot of these, this is only for C. elegans, which is free living, means they only feed on E. coli, but there are a lot of parasitic nematodes. Like if you look at the statistics, around like 1 billion people get infected by nematodes every year. So these nematodes that goes into like this roundworm, tapeworm, they are all basically nematodes. They do not infect their host when they are in this happily growing life cycle. When you starve them with food, they go into this hibernation type, this infectious state, and at this stage they behave this, show this typical different behavior, and they infect host. So this is a very common principle of nematodes. Yeah.
How do they know when to perform this mutation behavior, since all of them are doing it at once? So what cue is there?
So it's a good question. We don't know actually, because they do not do it all the time, but I think they just stochastic maybe, I am not sure. But in case of parasitic nematodes, people have actually looked at it, and they found that sometimes when the host cue is present, they do not only do that trigger nictation. The host cues trigger nictation for parasitic nematodes. Sometimes they not only do nictation, but they actually can coil up and jump towards the host cue source. Yeah, so for free living animal, I am not sure. Yeah.
Leeches also do this kind of
Leeches are considered a completely different
Animal, but I am not sure. I don't know.
I saw it myself just a week back.
Yeah, they actually.
And they actually stand on their end and like.
I think there's a similar behavior. Yeah. Actually, you're right. Yeah, I don't know whether there is any similarity. Circuit is similar or not. I don't know.
They would do that even for their regular locomotion, right? They do this.
They do like sometimes like. Yeah, I don't know whether there is any particular cue or anything. I don't know. Yes, I was just getting to it. So people have done that. Is again the gap junction information, electrical synapse information is not there. The chemical synapse information is there. So what is the main theme that came out? That if you look at the basic architecture of the network, it remained the same. The 90% of the chemical connectome remained the same, but now you have mostly some of these interneuron connections are getting stronger, and these new connections that you see in these ones, this is a command interneuron that generates forward locomotion, and this interneuron is connected to the command interneuron, but not here, because the locomotion has to be altered. But if you look at like how I am going to connect to this muscle skeleton. This command interneuron to the motor neurons are connected to the muscle skeleton. That is not changing. So, those levels are staying the same. The sensory neuron level also stays the same, but the interneuron level, they have lot of new connections and some loss of connections here. So, again I think I will stop here. So, again like why as I mentioned that there are modularity or circuit information is there. So, the next question we can try to understand is so that how physiologically conserved circuits or modules emerge, because the modules or circuits does not necessarily include neurons that are born together or like there is any commonality between them. So, many circuits they have like interneuron, sensory neuron, motor neurons together, they are performing like one particular task. So, we will like next tomorrow I can just talk about like how these circuits emerge. Okay, any other questions? Yeah.
Between the hermaphrodite and the male, is there some kind of repurposing?
Yes, there are some repurposing. It's not quite evident at this resolution, but if you look at like if you look at individual circuit level, this information is there in the connectome, but there are some repurposing. But what is surprising is like there is in many cases in higher vertebrates, there is a lot of synaptic pruning that goes on in during development or during reproduction, like sexual maturation, this puberty. Here in invertebrates, there's some pruning goes on, but not a lot. So, mostly is new synapse formation, but there are some repurposing means like that the neuron is still shared between two sexes, but it does something else. Yeah, that happens in males.
What stage is it determined whether it will become male or female? Male or female?
At L4. So, before L4, if you look at like if you look at the hermaphrodite circuit and the male circuit, we do not have the entire connectome for that, but some people have done like small circuit analysis. If you look at the same circuit in hermaphrodite and male, before L4 is like very similar. After L4, when the sexual hormones like comes in, you have like these circuits like diversify. Yeah, some you get some new synapse formation in males, because the new neurons come in males, around 90 neurons come in males, you have to accommodate them, and also some pruning goes on in the pre-existing hermaphrodite circuit.
Is the pruning being less correlated to behavior being more analog rather than?
Could be. We don't know that. We don't know that. One thing could be that it's so small and it's less wasteful in a sense, like but that's just a little just a hand waving. We don't know like why there is less pruning, why there is yeah. You do see some of the synapses. One problem with the connectome, these connectomes are that these are like very less individual. We don't have that many sample size. So, and the other problem is you cannot follow a single animal over developmental steps. Okay, so that is a problem. So now what other people have done, so they have devised some genetic tools to label precise synapses. Okay, and then you can follow the animal. You can image them like it's mostly microscopy based. So you can image them in the earlier stages, and then you can image the same animal in the adult stages, and you can ask the question whether it is staying the same or how it is changing. And in there you can see that there are some changes in the synaptic volume, the number of synapses is much more clear there, but we do not have those information at the whole circuit level. If there is no other questions, then thank you all.
Thank you so much.